This is Mitosis by Anirudh Kasturi and Pranav Rajan. So our first step is our interphase. Interphase isn't exactly a stage of mitosis. You can't see any of the individual chromosomes. It includes your G1, your S, and G2 phases. It is pretty much your normal stage of what most plants and animals go through on a normal basis. Your prophase is when your nuclear membrane begins to disappear, which is being absorbed by your cell, and the chromosomes begin to condense. Yet you can't exactly say that they're separated because they're just trying to get organized, and they are at the center of the cells. And the centrioles move to the poles and produce fibers called spindle fibers. Metaphase is when the sister chromatids line up at the equator and they attach to the spindle fibers through the kinetochore. And each sister chromatid is attached to a certain spindle fiber, which are then attached to the centrioles. Then we have our anaphase. Anaphase is when the centrioles pull the sister chromatids apart from the nucleus, from the center, and they start pulling them to the poles. And then the centromere also is breaking in this stage. And then our last stage is telophase, and in combination with cytokinesis. So in telophase, your chromatids have reached your poles, and the cells begin to divide. Cytokinesis can also overlap with other forms of mitosis, and the nuclear membrane also begins to reform, and there's now two nuclei. In cytokinesis, animal cells and plant cells divide differently. In animal cells, they pinch from the outside called cleavage furrow, while in plants, they form a cell plate and it expands and then it separates.